Greetings everyone, we are from Science Marine program. Today we would like to share with you guys our research methodology and scientific writing entitled Sea Turtle Slaughter. And this is our team. We are consist of 10 people. And for our contents, first we have introduction, research background, research objective, literature review, methodology, result discussion, and lastly conclusion.
About the introduction, illegal trade and habitat threats jeopardize sea turtles. Despite conservation, poaching on unprotected beaches devastates over 90% of nests. The global demand for eggs, meat, and shells intensifies the crisis. Legal consumptive use raises concerns. This study aims to locate illegal poaching and assess its impact on endangered species, crucial for ecosystems and local communities. Next, the research background. Pulau Tiga, Malaysia faces a crisis with sea turtle slaughter disrupting marine ecosystems. Human activities and inadequate conservation worsen the issue. Genetic studies trace poaching origins and hotspots. Researchers analyze poached turtles' fingerprint for targeted conservation strategies, vital for sea turtle survival and preserving ecological integrity in the region. After that, research objective. To determine the specific location at Pulau Tiga, to conduct genetic studies on sea turtle population in the region, to assess the magnitude of sea turtle poaching, and the last one is to discuss the potential consequences of this mass sea turtle slaughter. For the literature review, sea turtles globally face endangerment due to deliberate harm, particularly in Malaysia, Philippines, and Mexico. Studies reveal alarming trends like 77% adult and 23% large juvenile green turtles found dead in Pulau Tiga, Malaysia in 2014. Other research emphasized sustainable livelihood in the Philippines and mortality rates in Mexico. These studies emphasize the urgent need for conservation in the face of rampant illegal activities, stressing the importance of balancing social progress with environmental preservation. Next, we have three methodology. The first one is turtle carcass collection. Conduct interviews with local fishermen to identify areas of turtle poaching. Survey Pulau Tiga, identifying and collecting 53 sea turtle carcasses. Determine species, measure dimension, estimate maturity stage, and check for identifying markers. And retrieve all carcasses and sample genetic material for analysis. For the second methodology is laboratory genetic analysis. Extract genomic DNA using the CTAB protocol, apply PCR protocols to amplify mitochondrial DNA segments, and verify DNA samples via gel electrophoresis and outsource sequencing. And the last methodology is molecular and mixed stock analysis. Identify haplotypes using genetic databases, estimate natal origin using Bayesian mixed stock analysis, employ Markov-Chain Monte Carlo chains for analysis convergence, conduct mixed stock analysis using various priors, and assess contribution from neighboring foraging grounds through MSA and exact tests for haplotype compositions.
For our result divided into two, which is species identification and size frequencies, and the others is haplotype composition of carcass sample. For species identification and size frequencies, the total of turtle carcasses is 53. Curved carapace length is 67.0 to 111.0 cm. The mean is 91.3 cm. The standard deviation is 10.2 cm. Species old green turtle, which is identified on site. Tagged carcasses, 3 carcasses had tagged 2 from Turtle Island, Malaysia. Tag numbers MYS at 7117 and MYS at 7118, and 1 from the Philippines, tag number PH1526, and the age of the turtle is 77% adult and 23% juvenile. For haplotype composition of carcass sample, for the haplotype, 39/53 of genetic analysis successfully amplified, 10 haplotypes were detected. 8 haplotypes correspond to green turtle from nesting and foraging around Central from South Asia. 2 were registered in Uganda. Natal origin, Sulu and Celebes, which is median 53.0%, uninformative prior, to 0.1.3% informative prior, and Malay Peninsula and South China Sea, which is median 36.4%, uninformative prior, to 24.4%, which is informative prior. The MSA result significance specified that carcasses sample originated from the Turtle Island in the Sulu Sea. The estimated source foraging ground in Southeast Asia indicated a very high contribution from the Brunei Bay foraging ground, which the median equal to 90.2%, while Tun Sakaran Marine Park and Sipadan ground had relatively small median, which is 4.6% and 1.2% respectively. The haplotype composition of carcass samples was significantly different from foraging aggregation of Sipadan and Tun Sakaran Marine Park. However, it is not significantly different from Brunei Bay. For our discussion, it is also divided into two parts, which is the negative effect of sea turtle slaughter, and the others is the measure can be taken to prevent sea turtle slaughter. So firstly, for the negative effect of sea turtle slaughter, it can be categorized into four, which is decrease in nesting population, disruption of the natural balance, thirdly potential genetic impacts, and lastly significant threat to nesting population. For decrease in nesting population, sea turtle slaughter in the Sulu and Celebes seas contribute to a decline in the overall nesting population. As turtles are killed for their eggs, meat, or other body parts, the reproductive capacity of the population diminishes, hindering their ability to sustain and grow their number through successful nesting. And for disruption of the natural balance, the systematic killing of sea turtle disrupts the delicate balance within marine ecosystem. Sea turtles play crucial roles in maintaining the health of the ocean by regulating jellyfish population and supporting biodiversity. Their removal from the ecosystem can lead to imbalance, affecting various marine species and consequently the overall health of the seas. For the potential genetic impact, continuous sea turtle slaughter poses the risk of genetic impact on the surviving populations. The selective targeting of certain individuals for the widespread killing of turtle can reduce genetic diversity. This narrowing of genetic variation may compromise the species' ability to adapt to environmental changes, making them more susceptible to disease and other threats. Lastly, significant threat to nesting population. The culmination of this negative effect emphasizes the grave threat that sea turtle slaughter poses to nesting populations. The combined impact of reduced numbers, ecological imbalance, and potential genetic consequences underscore the urgent need for conservation efforts to safeguard these endangered species in the Sulu and Celebes Sea. And for the measure that can be taken to prevent sea turtle slaughter, first is enforcing laws and regulations that prohibit the killing, harvesting, or trading of sea turtle and their products. In Malaysia, sea turtle protection is covered by the Fisheries Act of 1985. States have their laws too. In Sabah, selling turtle eggs is illegal with a fine of Rm 50,000 or 5 years imprisonment. Keeping turtle products result in a fine of Rm 100,000 or 3 years imprisonment. Secondly, reducing the bycatch of sea turtle in fishing gear. Bycatch is accidental capture of non-target species harming animal population. Turtle excluder device TED and circle hooks in longline fisheries help reduce turtle harm, with TEDs cutting mortality by 97%. Thirdly, establish and strengthen protected areas around nesting beaches. Protected areas like Tunku Abdul Rahman Park in Sabah conserve biodiversity. They offer legal defense and safeguard turtles from threats like poaching and habitat damage, with programs for monitoring and rescue. Lastly, education and awareness to local communities and tourists about the importance of turtle conservation and the threat turtles are facing. For this, effective public awareness initiative seeks to shift the prevailing perception of turtle, reframing them from commodities to species worthy of protection. Organizing and conducting outreach activities such as exhibition and can increase the community's knowledge and awareness of the importance of sea turtle to the ecosystem.
So we conclude that the research underscores the severity of sea turtle poaching. Genetic tests pinpoint poaching hotspot and highlight risks to turtle populations. Urgent action is needed involving stricter law enforcement, awareness campaigns, and global cooperation. The genetic data not only deepens understanding but also guides future research. The study urges collaboration among communities, environmentalists, and legislators to halt poaching, preserving sea turtles and their habitats for a sustainable future. And this is our QR code for reference. And that's all from our team. Thank you.